Hi there. I will be explaining cryopreservations in plant tissue culture. I will be explaining these in details. What is cryopreservation? Plant material to be conserved in cryopreservations, the merits of cryopreservations in plant tissue culture, and cryopreservation procedures. What is cryopreservations? Cryo means frost in Latin, and cryopreservations means storing plant material. Since it is about plant tissue culture, therefore it means storing plant materials under freezing or very low temperatures. The temperature at which the plant material can be stored ranges from different temperatures. Either it can be in solid carbon dioxide, which has a temperature of negative 79 degrees Celsius, or it can be in deep freezers, which has deep freezer is a very big freezer that can freeze to a low temperature of negative 80 degrees Celsius, and the temperatures can also differ. It can also be in liquid nitrogen medium, which has a temperature of negative 196 degrees Celsius. In dealing with these substances or liquids and solids, you need to take some safety precautions like putting on a cryo gloves. The cryo gloves will help prevent you from getting frostbites. You get frostbites when you touch the dry ice or liquid nitrogen with your bare hands. Therefore, this cryo gloves is very important. Nitrogen is also a cryoliquid. Nitrogen can be in two phases, in vapor phase and liquid phase. The vapor phase is at a temperature of 150 degrees Celsius. The liquid phase is much more colder at a temperature of 196 degrees Celsius. Cryogenic liquids these are gases that are liquefied and maintained at very low temperature. Plant material to be conserved. This includes recalcitrant seeds, vegetative propagated species, and many of these that I will be mentioning a little bit later. Recalcitrant seeds. These are the seeds that cannot withstand very low or high temperatures. If they are exposed to very low or high temperatures, their viability reduces and they can desiccate, like they can dry out, therefore it will render them useless. And they can also suffer from chilling damages. This is an example of a chill damage of a banana. Therefore, because these recalcitrant seeds, for example, are very much prone to extinction, that is why their pods are kept in cryo liquids or cryo preservation procedures. Vegetatively propagated species do not have seeds, and even if they have seeds, it is usually heterozygous. So if the scientists want to have a homozygous, that is the original 100% of the plant's species, therefore the scientists need to take the vegetatively propagated parts of the species and store it in the cryopreservation way. Rare and endangered species are also stored in the cryopreservation procedures because rare and endangered species are the species that are facing extinctions because of certain conditions. So they are stored in order to keep their progeny from being extinct. And biotechnology products, which are usually very rare, are also kept in these procedures. Disease-free plants can be kept because plant species can face disease which might lead to the extinction of that particular species of plant. Therefore, if they are kept in the cryopreservation, it people or scientists can get back to them anytime they want to repropagate them at anytime they convenient for them if the disease is no longer into existence. Merits or advantages of cryopreservation. When a sample or a plant material is stored in a very low temperature, the metabolism and cell division of the cells stops. So if the metabolism and cell division stops, it means the plant cannot age easily, because in order for a plant cell to age, there are certain chemicals that leads it to age, or certain enzymatic procedures. At this temperature of maybe, let's say in liquid nitrogen, the temperature is cold, and because of the cold temperature, the metabolism and the cell division are frozen, then the plant can be saved. Indefinitely, because the metabolism has stopped, so the plant can be stored for any period of time, let's say for example a century, because the cells are not aging, and there is no chance of new contamination, because the liquid nitrogen is very cold that even psychotroph bacteria, which are bacteria kept that can grow or thrive at a very low temperature, cannot even continue to live at a temperature of negative 196 degrees Celsius. And the plant material that are kept are usually just occupy a small space, and therefore it is easier because of the small space that is used. There is minimal labor in trying to control them or maintain their viability in the cryo state. Plant material selection procedures. These are the most important procedures in cryopreservation, such as plant material selections. The plant material selection depends on the scientist and what the scientists want to store, or the particular sample which is facing certain environmental condition which force people in that origin to save those kind of plants, such as the plants that are about to extinct. Cryoprotection treatment. This is done by adding certain chemicals such as sucrose and dimethyl sulfur oxides, which help to maintain the plants in the cold state by protecting it from completely freezing. The cryoprotective treatment counteract the freeze harm that can be caused to the plant when it is freezed at low temperature. Storage. The storage can differ from the plant species that will be stored, and in storing it, it can be done by slowly freezing or rapidly freezing the samples. And thawing simply refers to defrosting the samples or the sample vials, and then it can be washed to remove the cryoprotective treatments or cryoprotective chemicals, and then it will be recultured to measure the viability. From there, it will be regenerated. Plant material selection, as I said, can depend. It can be any plant material that requires preservation in order for it not to extinct. Before protecting a sample from in a cryo state, the water content should be completely reduced by adding certain chemicals such as dimethyl sulfur oxide, alcohol, glycol, and sucrose, and many other chemicals. These chemicals counteract the sample from cryo destruction. and it is usually preferable to use two in combination of these chemicals rather to one, because it is believed to be much more effective than when you use one. In a case whereby a sample to be stored has a high water content material, the water content of the sample is very high, the water content should be reduced by any means to a very, very low level. Or if it is not reduced, when it is being stored in liquid nitrogen, it can face some cryo complications, such as the death of the cells. Storage. In storing samples, it depends on the plant species and is sensitive, because different plants have different sensitivity to low temperatures. It can either be in slow freezing method or rapid freezing method. In slow freezing method, the sample is slowly freeze at a steady rate, gradually to a higher low temperature, from lower temperature to the higher. And because it is freezing, the freezing is steadily, the ice crystals that are formed are very, very large, and they can damage the cells of the sample. And the slow freezing method, it kills only few pathogens, and therefore it is not advantageous compared to the rapid freezing method. The rapid freezing method, the sample is freezed at a very short period of time. Therefore, the ice crystals that are formed are very, very small, and therefore it causes a less damage to the sample that is stored. And this method, it kills almost all the pathogens compared to the previous one, which is the slow freezing method. It kills many of the pathogens that can cause complications. Storing the sample can depend whether it is going to be for a brief storage or a prolonged storage. If it is to be a brief storage, like a short period of time, the sample should be stored advisably in negative 80 degrees Celsius in a deep freezer, or in dry ice, which has a temperature of 78.5 degrees Celsius. For cryopreservation to continue, the sample or the tanks of that is the dewars. The dewars are the containers in which the liquid nitrogen is stored. The content of the liquid nitrogen should always be refilled in order to maintain the sample at that negative 196 degrees Celsius. After, when you want to propagate or regenerate the plant sample that you stored in in the cryo state, the sample will be collected and plunged into a water bath. With the water bath, it's usually at a temperature of 0 degrees Celsius, and in thawing it, the process should be very, very quick. Or else, if it is not quick, when the temperature reaches a certain level, the dimethyl sulfur oxide can cause damage to the cell membrane of the cells. Therefore, the washing is done to remove these dimethyl sulfur oxides and many more cryoprotectants. And then it will be recultured in a non-selective media, growth media. And because the samples are stored in a very low temperature, there are possibilities of storage lost because of death. And after this, the samples are then regenerated. When the viability test is done, they are regenerated to see which of the samples is viable and which one is not, by culturing it in a growth media, specific growth media, and the growth media should be in suitable environmental conditions of certain temperature and media for the samples to propagate